Part 2. An example of additive gene action will be the milk production of Holstein cow, at which each parent contributes to the overall milk production. Another example is fowl will be gameness or cutting ability, since both male and female contribute to the final effect. One of the great problems of the breeder is that the most important traits one is seeking to set in his line are not unifactorial, that is controlled by just one set of genes. On the contrary, most of the traits we are interested in will be affected by many genes or gene systems. These traits are known as polyfactorial or quantitative in nature. For example, the type of cock is determined by one set of genes. A pecan fowl will be dominant over a single cow. However, there is no proof that the type of cow make for a superior chicken. On the other hand, the gameness of the game cock is of vital interest to us. Excuse me. But since there are many genes involved in this trait, it is not easy to set at it should be to get a single cow family. We know we are dealing with the quantitative traits when there is continuous variation in the animal produced. Since we know that the game fowl range in degrees of gameness, we can assume we are dealing with a quantitative traits. To continue with this topic, we will have to discuss other types of non-additive gene action which are vital to the breeder. One type of gene action is considered to the lack of dominance. An example of this is when they breed a black strain to a black plus white strain, the offspring will be blues. This is an example where neither gene dominated over the other, or the gene seems to combine to produce something new. However, when these offspring are bred among themselves, they will produce blues, blacks, and whites. Another type of non-additive genes action is over-dominance. In this type of gene action, the offspring is superior for a given trait over either parents. For example, there are corn plants which, when bred, will produce progeny which are much taller than other parents, as much smaller than either parents. Okay. Another type of non-additive genes action is known as epistasis. The key to epistasis is based on understanding that most traits are determined when two alleles, that is genes on corresponding section of the chromosome, fuse to form the new individual. For example, a chick inherits a gene for yellow leg and one for green leg and is born yellow-legged. However, there may be a gene on another chromosome altogether which modifies these two genes and produces a fowl with yellow leg with large off-color splotches. A very interesting epistatic effect in fowl has to do with the way in which white feather color is inherited. There are two types of white in fowl, a dominant and a recessive white. However, there is also a gene in fowl which inhibits feather coloring. This fowl is also white, although it may include color in its genotype. When white leghorn are crossed with white Wyandottes, all the progenies are white. However, when these fowl are bred among themselves, you will get many colored chicken. The reason is that the leghorn is actually a colored bird, but also homozygous. For the inhibitor, since the inhibitor gene is dominant, in the first filial generation, all fowl obtain at least one dominant, one gene, and one recessive gene. As a result, all are white. In the second generation, the fowl that do not obtain the dominant I gene will be able to express the color that was always there in the Leghorn strain. Again, although color is an interesting trait, it is not vital to the breeder. Although the above explanation may help you understand why you breed whites and got reds. More importantly, epistatic genes, however, affect the overall health and vigor of the animal. The above explanation, although technical, is vital to the breeder because it helps him to understand how his fowl may be inheriting specific qualities. In summary, we can state that the majority of the important qualities we are interested in are quantitative, cutting, gameness, and action in nature, that in additive in effect, while many of the lesser important qualities, color of feathers and legs, comb type, eye color, egg color, and non-additive in nature, although there are exceptions to put into perspective just how important these concepts are. We would like to summarize the breeding implication of the above. If the trait under consideration is determined by polygenic additive traits, it is moderately to highly heritable. It will be little affected by either crossbreeding or inbreeding. Traits affected by non-additive traits will usually has low heritability and will show a decline when inbreeding is practiced. If a trait is low to medium in heritability but shows improvement in crossbred, then it is probably affected by both additive and non-additive genes. Non-additive genes action is responsible for that sought after quality called the hybrid vigor. We have hybrid vigor when the offspring are better than the average of the family. Should the offspring be better than either parents, then we are dealing with over-dominance. There are the famous nicks we are always hearing about. The basic problem of the breeder can now be posed and possibly even answered. How can we breed our fowl in such a way that we can take advantage of both an additives and non-additive gene action working in our breeds? That's the question. If we can answer this question properly, we will be on our way to an ever-improving program with ever better fowl. In order to take advantage of non-additive genes action, we must bring together animals with different genotype so that the differences in the genes or alleles have a chance to express this type of action. Non-additive genes action is based on differences in the genes being brought together. In order to take advantage of additive genes action, we must bring together similar genes so that we can build upon the similarity. Also, we must always keep in mind this type of gene action and intersection work in contrary direction and actually conflict with each other. This is especially so when we realize that there are thousands and thousands of genes in our fowl acting in these two contrary ways. We must develop a breeding plans which take this into effect and establish a balance between two different ways of inheriting traits. That's part 3.